Welcome to the third lecture for the respiratory system for Biology 153. Before I start, I just wanted to do a little bit of a recap. The first thing that we had been talking about when it came to respiration to explain how breathing occurs is we talked about ventilation, bringing the air actually into the lungs. And you can see here that the first step is going to be muscles, expanding the thoracic cavity, and those muscles are these ones that we're talking about here, the external intercostals and the diaphragm. They will contract to expand the thoracic cavity, but just because thoracic volume increases wouldn't necessarily mean that lung volume also goes along for the ride, but it does. Lung volume does increase because of the transpulmonary pressure gradient. And you can learn more about the transpulmonary pressure gradient on the first page of the ventilation notes. Now that lung volume has increased, we also note that pressure in the alveoli will decrease. This is Boyle's law. And since we have a difference in pressure, we now have a pressure gradient, and gases will flow from high pressure to lower pressure, or we could say gases flow down the pressure gradient. And at this point, you could actually insert all of the respiratory anatomy. So it might, for example, go in through your nasal or oral cavity, and then down the trachea, past the oropharynx and laryngopharynx, of course, into the trachea, and from the trachea through the bronchi, bronchioles, all of the different little bronchioles like terminal bronchioles, respiratory bronchioles, and on until we get to the alveoli. Once we get to the alveoli, now we have to take a look at how the oxygen gets, or the air gets, from the alveoli into the blood. And so this is the next main topic that we were talking about, external respiration. Here you can see oxygen is going to be diffusing into the bloodstream, but when oxygen diffuses into the bloodstream, that diffusion depends on the partial pressure gradient, the difference between the pressure of oxygen in the alveoli and that of oxygen in the pulmonary capillaries. Being able to diffuse into the pulmonary capillaries also depends on the solubility of oxygen. So once it actually enters the capillaries, it has to be able to dissolve in that fluid. And that depends on two things: the partial pressure of the gas, which can be changed, and the solubility, which cannot. So then, once a certain amount of oxygen has actually diffused into the bloodstream, now it has to actually get into the red blood cell and be transported by hemoglobin, because oxygen is not very soluble in fluid, in water. So that's really the next note that we're taking a look at, how oxygen is transported by hemoglobin. Just a recap of this as well. When you think of hemoglobin, I almost would like you to think of it as a coin purse, so that, for example, here you can see there's 4 different areas. If I go back to that picture of hemoglobin, you can see here we have the globin subunits, 2 alpha and 2 beta, and then within the middle of that we have this green disc, which is the heme group, and at the center of the heme group we have iron, where oxygen binds. So when you think of this hemoglobin, note the 4 heme groups, the 4 iron, the 4 possible area where oxygen binds. So if we take a look at my analogy, the coin purse, it's almost like in this area here, you could have iron where oxygen can bind. And the way hemoglobin works, there is cooperative binding between oxygen and hemoglobin. That means if one molecule of oxygen binds to one iron in hemoglobin, that makes it a little bit easier for the next one to bind, because that one binding kind of opens up hemoglobin, changes it from the tensed position, the tensed form, to a more relaxed form. And you see, once the first molecule of oxygen binds, it makes it actually a little bit easier for the second molecule of oxygen to bind. And once the second one binds, pretty much the whole hemoglobin just flips open, and then it's really easy for all 4 oxygen molecules to bind. And so we say it is in its relaxed state, where it's really easy for hemoglobin to pick up or become saturated with oxygen. So going back to our notes for a moment, when we graph this cooperative binding, what we see is a graph that looks like this. And you can see in this part here, what we're showing is if we are taking a look at different levels of PO2, here are the different levels of PO2, then depending on the different levels of PO2, then hemoglobin will be more or less saturated. So when there's lots of oxygen around, you can see hemoglobin is well saturated, maybe 98% saturated. And because this curve is an S-shaped or sigmoidal curve, then even if initially you have quite a big drop in hemoglobin, like here 40 millimeters of mercury, there's not much of a drop in hemoglobin saturation. Hemoglobin still holds on to oxygen. And that's really great because let's say at sea level PO2 is 100. If you were to go up to the top of Whistler or go skiing for the weekend, PO2 is likely to drop not more than, say, to PO2 equals 80. And if that's the case, you can see hemoglobin still picks up oxygen really well. It's over 94% saturation. So that means you can go skiing or snowboarding for the weekend and really not worry about being able to breathe. On the other hand, if we look at the other end of it, here it's very steep. So here, a small drop in PO2, like if your resting cells suddenly are a little bit more active and they use up a bit more oxygen, then hemoglobin will drop off a lot of oxygen to them. So it's really very handy.